Is it good? Okay, so I'm Luigi. I will present the work that I did in the lab of Ruben Portuguese in Munich at the Technische Universität. And I will talk about what I've provocatively called a cognitive map that we think we can observe in a brain region of the larval zebrafish. So I'm a neuroscientist, not an ethologist. There's no cool animals in my presentations. But you can put your animal of choice as this triangle. And you can think that as it moves through the environment because of foraging, exploration, or navigation, like migration, this animal needs to somehow keep a constant representation of the direction that it's facing to know where it's heading toward. And like people have been thinking that like the best way of like represent this efficiently in the brain is in so-called ring attractor networks. Networks that we can think about as like neurons that we can conceptually organize in a circle, and neurons that are in different parts of this like theoretical circle will code for a different direction where the animal, when the animal is moving. So as long as the animal is pointing toward one particular direction, one part of this network will be active and constantly active, and all other neurons will be silent. And as the animal then changes the direction, the activity in the network will propagate clockwise or counterclockwise, keeping as a compass a constant representation of the direction the animal is moving toward. And like the most beautiful example of this kind of network has so far been described in the Drosophila. In the Drosophila central complex, there is a region that is called the ellipsoid body, which is approximately a circle, and that's why it's so cool. Because if you look at activity in this region, in a fly that is like head fixed on a ball, so the fly can navigate as like defining navigation as like moving left and right on this ball. And there is a virtual arena, so there is a bar that moves left and right as in a virtual environment. There is a bump of, in this case, calcium activity that propagates one direction or the other in the ellipsoid body as the animal moves left and right. So just by looking at where activity is in this region, we can read out the fictive virtual orientation of the animal in the virtual environment. And the very good thing about Drosophila is that like this was described 7 years ago, but like there is a lot of genetic access and electron microscopy reconstructions that are allowing people to understand this circuit really at an incredible level of detail. But there is a question standing that is, can we find a network similarly arranged to code for heading direction in vertebrates? Because in vertebrates, you all know that like there's place cells and head cells, head direction cells and grid cells that have been described, but so far there is no evidence of an anatomical organization for the heading direction network. So we have been working with the larval zebrafish, which is a pretty unimpressive animal itself, but it's very cool because it's super easy to breed, it's small, and it's transparent. And also it's genetically tractable, so we can have larval zebrafish that express in all neurons of the brain or a subset of neurons in the brain fluorescent proteins that we can use for reconstructing the anatomy or also to look at activity in a behaving animal. And I will show you data that we acquired with Lightsheet microscopy, which is a technique in which we can head fix an animal in jelly under the microscope, and then we can look at activity at basically every brain region, and like you can see this movie, where I'm showing you calcium activity from the whole brain of the larval zebrafish as it moves under this setup. And at the same time, we can put a camera and we can look at this guy. He's pretty chill, sometimes move one direction, the other, or forward. And we can reconstruct the position of the tail, and we can look at when the fish is moving one direction or the other direction. So our observation started with like imaging a specific line, which is a line that is expressing the calcium indicator that we use to follow activity, not in every single neuron of the brain, but just in a fraction of inhibitory neurons of the brain. And what we observed was that there was a region of the brain, which is called like the anterior hindbrain, where we can find those two clusters of GABAergic, so inhibitory cells. And the very cool thing about this cluster is that when we actually went and looked at activity there, as the animal was moving, we could observe something like what you see in the movie. So basically a bump of activity that was propagating in one direction or the other, as all the rest of like neurons around were just. So if we just isolate those kind of neurons with a very stable pattern of activation, you can see how it seems that there is a bump of activity that is rotating leftward, like clockwise and counterclockwise on this network. And we can use mathematical techniques similar to what Ali just presented to organize those circles, those neurons based on their proximity in functional space. And we can use this kind of representation to basically derive, doesn't matter how, a vector that is pointing at every point in time in the direction of maximum activation of this network. So we can now have a single variable that can capture over time which part of the network is the most active. So in the plot on the right, I'm showing you activity over time for all neurons sorted according to their position along the circle. And you can see that the bump of activity that you see in the movie, you can see it as like this translation, this white stripe that moves up and down in the plot. And the green line is this phase of the network that is constantly tracing where the peak of activation is. So now that we have this quantification, we can actually ask the question of what is driving those changes in the network. And you won't be surprised. What we need to look for understanding this activity is the motion of the fish. And now in gray, I'm showing the trace of the tail. And from this trace, we can we can find ways of reconstructing where the fish was going left, when the fish was going right, and when the fish was going forward. And you can see that the part of the experiments where the phase was changing the most were points in time where the fish was moving more strongly in one direction or the other. So what this means is that if we, similar to what was happening in the Drosophila, if we calculate a virtual heading of the fish in a virtual space, this virtual heading correlates very well with the phase that we can read out from the network activity. And actually, this is just one animal, but this was very consistent over many animals. But another very cool thing about this little guy is that also for larval zebrafish, we start having electron microscopy data basically coming from the whole brain of the organism. So we have been using 3D electron microscopy data set that encompasses basically the whole brain of the larval zebrafish. And in this data set, we can look at neurons with a level of details that allow us to resolve individual synapses. So we have been reconstructing cells of, in this region of the brain that we were also imaging. And you can see here in this video that when we go looking into this particular brain area, and it is the anterior hindbrain, but more specifically the interpeduncular nucleus, we can find that there are those neurons that we call R1 pi neurons that project, like that have their soma dorsally, and they project dendrites and axons ventrally. And if we look in more closer detail, like this is just the frontal view and the horizontal view of those neurons. And if we look in more closer detail about their morphology, you can see how they have a dendrite that extends in this, like downward in the same half of the brain, and an axon that is reaching contralaterally to the other side of the brain. And most importantly, there was a correlation between the position of the dendrite and the position of the axon. So a dendrite more on the right would correspond to an axon more on the right. And this was consistent over many neurons that we reconstructed. And why is this important? Because now we can actually think about this connectivity scheme, and we can observe how basically this way of, like this pattern of projection put in place a system where those neurons form recurrent connections with each other that are basically mapped linearly over the over the frontal axis of the fish, where the dendrites and their axons are in an off phase of 180 degrees. And we can postulate that this kind of offset that we see at the level of the processes correspond to the circular organization that we observe in the functional organization of their somas. And this would be the prediction, and when we actually look at the data, we can see that actually this sequence of coloring of dendrites and axons also correspond to the predicted sequence of coloring in the at the level of somas. So as I was saying, since those neurons, remember, are inhibitory and GABAergic, what this network can provide is the fact that when one part of the network is active, the opposite side of the network is completely suppressed. So this is a strict requirement for this kind of activity that we want to see, where there is a bump of activity that propagates and only one part of the network can be active at every given point in time. This obviously does not address the question of how this activity is moving around and how this activity is stable, which are further questions that we're trying to, people are trying to address. So to summarize, I show you that those neurons in the anterior hindbrain represent heading direction in the brain of the larval zebrafish, and that there is architectural principle in the organization that can somehow implement this kind of dynamics. And with this, I would like to thank all my group in the Portuguese lab, my collaborators, Hagar Lavian, who's who co-led this project with me, Ruben Portuguese, my supervisor, all the rest of people in the lab, the community that supports many of those analysis efforts, and you for your attention.
Thank you, Luigi, for your amazing presentation. It was really nice. I don't know if there's any question for the public. Yeah.
Hi, I think it was a really cool talk. I was wondering that your work explains how zebrafishes could navigate in a 2D world, the 360 degrees turning left and right. But I was wondering if there are other specific neurons that can explain the head direction with the inclination and like the elevation and depression, because they live in a 3D world, right?
Yeah, that's an amazing question. And like most people who work with larval zebrafish, including us, generally focus just on the 2D, but there's for sure a lot of complexity to be explained also in the 3D. It's very tricky because we at the moment cannot think of a system of tracking, of having a virtual reality in 3D. Like it's very easy to understand if the fish wants to go left and right just based on how they move the tail. It's very hard to predict how they would change their pitch. And so experimentally it's tricky because of that. But yeah, I think it's an exciting possibility for
Thanks. Also, a second question. When I was going through the CIMeC and Jayaraman paper that you showed, I thought that the torus was topographically the only surface that could have this ring attractor kind of connectivity. But your work shows that it's not a torus, but it still has connections that are similar to those seen in the Drosophila, right? So, does it mean that you don't need a torus?
Yeah, no, so yes, exactly. So when people think about those neurons have to be those neurons don't have to be anatomically on a circle. As long as those neurons they can be just scrambled in all directions, as long as there is a connectivity principle that connects them in a pattern that would match conceptually a circle. Right. It's just like I can imagine that having them on an actual pseudo-circular arrangement can make it very easy during development, just using gradient cues to organize those kind of projections in the proper way, in a way that is simpler than it would be more just intermingled configuration.
Thanks so much.
Any other question?
Thank you, Luigi. Couple of speculative questions. First, do you think there is some molecular marker to identify the cells that contribute to specific ring network?
Sorry?
Molecular markers that can identify specifically the cells that contribute to this specific network?
So this would be very important because at the moment we are lacking the tools for doing loss of function studies because like we have this line that labels those neurons but also neurons throughout the brain. So one of the things that we're trying to do is actually to look like in databases that exist of transgenic lines if we can find something that is specifically expressed in those. We have some candidates. We are trying them out. The problem is you can you have to image them first to be sure that it's those cells. So maybe it will happen.
And the second question, this is more philosophical. Do you think this kind of ring network is specific for like the head direction feature, or is a stereotype like microcircuit that applies to also other brain function?
So other great question. The interpeduncular nucleus that I was showing has two parts, a dorsal part and a ventral part. The ventral part I've not talked about, but there seems to be very similar connectivity principles, although the cells that go there are not the cells that we're looking that have like head interaction information. And they also receive, like this nucleus also receive from habenula, which is an upstream region, and the dorsal part and the ventral part receive from the left and right habenula, who convey probably very different informations. And this really raises the possibility that maybe similar organizational principles are just employed differently by like doing different tasks. Like I wouldn't call them necessarily to abstract, but yeah.
I have one last question, if I may. Why did you choose, before starting your experiments, to target the GABAergic population instead of any other population?
Okay, because the real story is the other way around, which is often the case when you in science. So we started from the interpeduncular nucleus, and we saw those neurons with those projections. They were like, okay, those projections are super cool, they must be doing something. And we had this GABAergic line, and we noticed that in that GABAergic line, there was very, you can see it, if you just look at the confocal image, you can see that there is very strong dark expression in those bundles that are going ventral. And it's why we started looking at this line.
Okay, thank you. So if there are no other questions, I think we can thank Luigi for his presentation. Now we will have the lunch break, so see you back here at 10 minutes to 2, I would say. So, yeah, see you later. Lasciane un po' anche a noi.
Io non ho i video degli animali carini.
Hai ragione, in qualche modo devi compensare.
Esatto.